Good morning, ladies and gentlemen. Welcome to the last session of talks for EEM 2023. I now invite the chairs for today's session to take their seats. Dr. Victoria Custodis, UKMAG, Switzerland. Dr. Milan Vukic, Faculty of Technology, Zvornik, University of Sarajevo, sorry. And Jelena Tanasic, Faculty of Technology, Novi Sad.
Good morning, ladies and gentlemen. It is our pleasure to welcome you all to this final session of the closing day of EEM 2023. As with so many good things in the life, usually the best comes in the end. So it is with the EEM 2023, and we saved the lecture of our esteemed Professor Susanna for the end. And without any further ado, I will kindly ask our Vesna to give us a short introduction and the main summaries.
Thank you, Milan. Susan Apostolov was born in 1984 in Bihać. She graduated in 2007 at the Faculty of Sciences, the University of Novi Sad, at the Department of Chemistry, Biochemistry, and Environmental Protection. She defended her doctoral dissertation entitled Study of Retention and Solvatochromic properties of newly synthesized phenylacetamide derivatives at the same faculty in 2015 at the chair of analytical chemistry. She became junior researcher in 2011, research assistant in 2012, and assistant research professor in 2015. Since 2017, she has been employed as an assistant professor at the Faculty of Sciences in Novi Sad at the Department of Analytical Chemistry, and in 2022, she became associate professor at the same faculty. She participates in the teaching of several subjects at the undergraduate, master's, and doctoral studies in chemistry: Analytical Chemistry 1, Analytical Chemistry 2, Analytical Chemistry Lab, Analytical Toxicology, Analysis of Biological Material, Preparative Analytical Chemistry, and selected chapters of Analytical Chemistry. In her scientific and research work, she deals with chromatographic examination determination and physical chemical characterization of newly synthesized, potentially biologically active compounds. So far, she published 21 papers in journals of international significance, 16 papers in journals of national significance, and she was participant at more than 50 international and domestic conferences. She is the author of a monograph of national significance and co-author of a collection of tasks. She was a participant in a bilateral project, and she is currently a participant in a national project financed by the Ministry of Education, Science and Technological Development of the Republic of Serbia. She was the mentor of several graduate and final theses, as well as a reviewer in recognized international journals. She is a member of the Serbian Chemical Society of Vojvodina. Let's greet Dr. Apostolov with a warm applause.
Good morning, everyone. Thank you for so nice introduction. First of all, I would like to thank the organizers for this opportunity to be the part of this traditional, international, and EEM Congress. And it's my pleasure and honor to be here today with you. As you hear, I talk about chromatographic parameters as predictors of compound biological activity. First of all, I would like to introduce you with some, what happened? Is it true? Okay, just battery, not me. Like I said, at first, I would like to introduce you some important numbers. We all know that drug development is expensive and lengthy process. According to some investigations, an average investment of pharmaceutical companies in discovering and bringing the new entity on the market in 2022 cost approximately $2.8 billion and takes around 15 years of development. Some predictions say that this amount will be even more than $5 billion until 2022. So why is the reason of so expensive and lengthy process? Namely, drug development is a very complex process. It includes 3 key stages. The first stage is discovery phase, which the goal is identification of relevant molecular targets and active compounds. The second one is development phase. Within these selected compounds enter the preclinical and clinical trials. And the last one is a regulatory phase, which enables approval, registration, distribution on the market, and post-marketing surveillance. An interesting fact is that about 10,000 preliminarily selected compounds, only 250 enter the preclinical trials, just 5 undergo clinical trials, and only 1 will be approved as drug and reach the market. How to choose from these 10,000 compounds those that enter the development phase? Well, until the middle of 20th century, drug discovery phase relies on traditional approaches of stepwise synthesis of potentially candidates, and you will agree that it is more easier to perform synthesis of compound with definite structure than compound with certain properties. So the greatest attention of contemporary researchers is focused on the steps which precede the targeted synthesis of potentially active biological compounds. Today, these steps are made easier thanks to computer-aided drug design, shortenly CADD. The main advantage of CADD relies precisely in discovery phase. But in addition, there is one more thing very important, and that is computer-aided drug design enables the identification of novel potential uses of drugs already approved for other applications. In pharmacology, we called it like repurposing. So how does CADD looks like. At first, we have to find target protein, and the question should be answered is, do we know its 3D structure? If we do, then we choose structure-based drug design. If we do not, then we choose ligand-based drug design. After that, we use complex screenings for all compounds that we know. And after that, we choose potentially the most promising series. We called it heat molecules. These heat molecules have to be proven by their selectivity, physicochemical properties, potential. And we applied it thanks to structure-activity relationship methods, which result is lead molecules. Lead molecules have to be improvements or optimized by several operations, which includes its iterative structure modification, an early in silico evaluation, and modeling of its pharmacokinetics, toxicity, and safety. Before extensive in vitro and in vivo experiments, and after that, we obtained drug candidate. But how to choose again those compounds? Well, we have to quantify the effect of structural changes on the properties of future compound, neither synthesized or still unsynthesized. So we have to try and find some help thanks to quantitative structure property relationships and quantitative structure activity relationships, shortened known as QSPR methods and QSAR methods. Among numerous molecular descriptors which are used for established QSPR and QSAR methods, lipophilicity is the most frequently used because it is closely related to the bioavailability of compound, and also it's important to pharmacokinetics, but also for pharmacodynamics and toxicity of future biological active compounds. So considering all of this, we want to evaluate, but first predict the existence of potentially activity of some novel derivatives, and we choose derivatives of phenylacetamides. Why these derivatives? There are many reasons, and some of them are the first one, that a quarter of all drugs in market and also 2/3 of all lead molecules are some compounds with amide functional group. The second reason is that history say that most popular analgesics in 19th and 20th century were acetanilide, phenacetin, and acetaminophen. The last was more familiar to us under the name aspirin. And the third reason is the most important reason, is that some contemporary researchers find that in addition to these analgesic properties, phenylacetamides show another and number of other biological properties. So we have that in mind, and we know that the type and intensity of biological activity of compounds is strongly connected with the chemical nature of presented substituent attached to the basic molecule. So we choose to examine 3 different groups of phenylacetamides, which differ in the substituent R1. Within the same group, these derivatives differ in the substituent R2. So we have 3 different groups of molecules: cyanophenylacetamides, chlorophenylacetamides, and diphenylacetamides. They are synthesized thanks to our colleagues from University in Belgrade, from Faculty of Technology and Metallurgy. And I say one big hello to them. Thank you for these nice derivatives. And I'll show you what was our first task. Our first aim was to examine whether these derivatives have some theoretical predisposition for good bioavailability. We checked their agreement with 2 drug likeness rules known as Lipinski's rule of 5 and Gauss's rule. All of molecular descriptors uncovered by these rules, we obtained by relevant software packages, which we find online. And we apply these rules for all examined derivatives, but I'll show you some results obtained for chlorophenylacetamides group. As you can see from this table and this table, the all examined chlorophenylacetamides show some probability to have good bioavailability in the organism because they do not show deviation in more than one criteria encompassed by these rules. Also, from this table, we can see that regardless to the mathematical approach, the high values of log P or partition coefficient we obtained for the compound with iodine as substituent. In contrary, the lowest log P values we obtained for derivative with most polar group, hydroxyl group, and we also can see that derivatives with the same substitution in different position do not show some differences in log P values. And another experimental is that in addition to these computationally obtained lipophilicity, we try to examine lipophilicity of these derivatives experimentally. And we didn't know which method should be used, but we know that behavior of compound in biological and chromatographic behavior are very conditioned by similarity in interactions of compound and their environments. So we decided to use reversed phase thin layer chromatography, and we used 2 stationary phases, 8 mobile phases, and after developing and drying and identification of compounds, we applied specific equation for chromatographic measurements, and we obtained 3 important chromatographic parameters. Chromatographic retention constants, RMO, chromatographic parameter M, and hydrophobicity parameter CO. On this slide, you can see the results of chromatographic measurements for chlorophenylacetamides, and you can see that high values of regression coefficient show that these RMP dependencies are very valid in the chosen work of experimental, in the chosen field of experimental work. And also we could see if we compare RMO values for different compounds in the same organic modifiers, that they are different between cells. So what's the point? Well, if we look at the same organic modifier, we could see that the higher values were obtained in acetone. And if we compare RM values for the same compound in different organic modifiers, we could see that, again, highest RMO values were obtained for the most lipophilic compound, the lowest for the most polar compound. And again, we didn't see any strong differences between RM values for compounds with the same substituent in different position. This confirmed our results obtained previously by mathematical approach. And in addition to all of these, we could see that M values follow the changes of RMO values. We supposed, we assumed that these parameters depends on the same physical-chemical parameters, so we correlated them by method of linear regression, and we obtained satisfactory linear correlations, even though in methanol we obtained 2 separated dependencies based on the polarity of substituent, and we confirmed that an protic and polar methanol is more sensitive than a protic and less polar acetone. So we examined lipophilicity mathematically, experimentally, and now we want to find some other correlations. In our previous research, we confirmed that RMO values are valid measurements of lipophilicity of cyanophenylacetamides group, but now we want to examine whether chromatographic parameter M, in addition to RMO values, could be reliably used as parameter of lipophilicity of chlorophenylacetamides group. So both of these parameters, RMO and M, we correlate with mathematically obtained log P values. And again, we obtained strong linear dependencies described by high values of regression coefficient R. And in this way, we confirmed that both of chromatographic parameters, RMO and M, could be reliably used as measure of lipophilicity of chlorophenylacetamides. As I previously said, it is very important on the start of research to examine the pharmacokinetics of future biological active compounds. So we decided to examine important pharmacokinetic predictors of chlorophenylacetamides group, and by relevant online programs, we obtained some important. They are shown in this table, and you can see that, that the highest absorption and possibility to bind to plasma proteins has the most lipophilic compound. In contrary, as was expected, the lowest absorption and the largest amount of unbound compound is described for derivative with most polar hydroxyl group. Also, we could see that derivatives with methyl group and halogen substituents could be used as neurological agents because of their values of log BB, and that values indicate that they could pass blood-brain barrier. I previously said that is lipophilicity closely related to pharmacokinetics, so we decided to previously obtained RMO and M values correlate with these important pharmacokinetic predictors, and we again obtain strong linear dependencies described by these high values of regression coefficient. And we found for the first time that RMO and M could be reliably used to assess the pharmacokinetic behavior of chlorophenylacetamides. In addition to regular used linear regression methods, we want to examine some other and deep interactions between chemical nature of substituents and bioactivity parameters of all examined phenylacetamides by applying methods of multivariate analysis. We used cluster analysis and principal component analysis. The results of cluster analysis for a group of chlorophenylacetamides are shown on this slide, and a dendrogram of bioactivity parameters show that all of parameters examined for chlorophenylacetamides are divided into clearly visible clusters. In first cluster, we can see that chromatographic parameter M is separated from all rest lipophilicity parameters, which are placed in second cluster. And the main results of this distribution is that chromatographic retention constant Rm is most similar, show better similarity with mathematically obtained log P values and pharmacokinetic predictors. Also, we applied cluster analysis for all examined derivatives, and we can see that all derivatives also divided into clearly defined clusters. In first cluster, we could see mostly of compounds with halogen substituents, and the other cluster includes all the rest compounds. But there's some separation within that second cluster, and we could see two subclusters. The first subcluster contains derivatives with polar substituents, and the other subcluster contains unsubstituted compounds, compounds with fluorine and methyl group. In addition to cluster analysis, we applied principal component analysis, and this analysis applied to the bioactivity parameters of chlorophenylacetamides results in loading plots. And we could see when we compare two main components, PC1 and PC2, that all of examined bioactivity derivatives are separated predominantly based on the value of PC1. Namely, PC1 separated these parameters based on the same distribution that we could see in cluster analysis. But thanks to small values of PC2, we could see that within the same group described by negative PC values, there are another separation of chromatographic parameter M from those theoretically descriptors, which is related to the nature and number of atoms. And we could say that although PC2 values are very small, PC2 in a finer way separates analyzed parameters based on the information they carry itself. Also, we applied principal component analysis on the examined chlorophenylacetamides, and as a result of comparing PC2 and PC1 and PC2, we could see score plots presented on this slide, and we could see that, again, PC1 predominantly separates examined derivatives based on the polarity of the substituent. This separation is similar to those obtained by cluster analysis, but there is one advantage, and that is that compound with methyl group is with those that are lipophilic. Also, based on PC2 values, we could see that compound with polar substituents are distinguished from those that are unsubstituted compound and compound with fluorine. We compared other main components, but we couldn't get any significant distribution of compounds. But when we compared PC1 and PC5, we find some interesting results. Namely, PC5 separates compounds with the same substituent in different position. So this is another advantage of PCA analysis, which confirmed and which reflected in their sensitivity to find some dissimilarities within the compounds with the same substituent in different position. But advantage is even more important because we couldn't get these separation in previously mathematically and experimental methods of determination of lipophilicity. In continuation of our research, we included another stationary phase, another molecular descriptors are included in our calculations, and we could find some interesting results for a group of D-phenylacetamides. You can see on this loading plot figure that by comparing PC1 and PC2 values, these parameters are on the same way separates as those as previously obtained in our researches. But there is another interesting thing, namely chromatographic parameter M show more similarity with toxicity parameters, and chromatographic parameters RMO and CO show again better agreement with log P values and pharmacokinetic predictors. But PC2, but PC2 did something better. Namely, PC2 values distinguished those parameters described by positive PC1 values into two groups, namely chromatographic retention constant RMO and hydrophobicity parameter CO are distinguished from those parameters of lipophilicity obtained by mathematical method. So we could say that advantage of PC analysis is another and reflected in its possibility and sensitivity to all those similarities between parameters, detect some dissimilarities between examined parameters. We applied PCA on the examined D-phenylacetamides, and we obtained separations similar to those previously obtained, but there are some interesting separations based on the PC2 values. Namely, I said that PC1 value predominantly separates analyzed compounds based on the polarity of the substituents, but PC2 values separates analyze compounds based on the electronic effects of presented substituents. So for the first time, we obtained these results, and we want to quantify the effect of polarity and electronic effects of substituents. So we correlate PC1 values with Hunch parameter as one of important parameter of lipophilicity, which describes individual contribution of substituents to the lipophilicity of whole molecule. And on the other hand, we compared PC2 values with Hammett constants, which describes the electronic effects of substituents. You can see that we obtained satisfactory linear relationships, and we found that all of these examined bioactivity parameters are most influenced by the polarity of the substituent, and to a lesser extent to its electronic effects. On the end, there are some interesting results related to all of these examined phenylacetamides. Namely, we want to find some relationships between presented R1 and R2 substituents on their bioactivity parameters, and we found, applying PC analysis, that based on PC1, all of examined derivatives are predominantly separate by their R2 values. But, sorry, PC2 separates all of these examined compounds based on the R1 values. I could say on the end, summarize our results, that we found that in the most extent and most large extent, bioactivity parameters of all examined derivatives are conditioned by polarity of presented substituents, and to lesser extent by electronic effects of presented substituents. And also we find that applied organic modifier, we supposed to say environment of compounds, in lesser extent conditioned all the bioactivity parameters of examined phenylacetamides. Now we deal with some in vitro research with our colleagues from Department of Biology and Ecology from Faculty of Science in Novi Sad, and we hope that in some future research we could find some interesting results by in vivo test of these phenylacetamides. Thank you for your attention.
Thank you, esteemed Professor Apostolova. I now open discussion. Are there any questions in our audience? Would you like to ask anything more? Do you want to ask? Yes. So we have a.
Yeah, thank you very much. It was very interesting. I was wondering, now that you have this correlation with the phenylacetamides, can you extrapolate this chromatographic methodology to other groups of APIs, or are you not? Is this?
Am I understand very well? I don't know. You ask me for other compounds, not only phenylacetamides, or?
Yes, I mean, you now developed this whole
yes, we
methodology,
try on
so-
azodides, derivatives of barbituratic acids, and etc.
So you were also planning to extrapolate this method to other groups of compounds?
Yes,
Oh,
yes.
it's very good. Thank you.
Are there any other questions? If there is no questions, I would like to thank you for this presentation, and I must say it, really exquisite presentation of your work. Thank you for coming here and giving us your latest results. Now we will proceed with the presentation. Yes. Of laboratory of Professor Murat Kaya from University of Basque Country in San Sebastian, Spain. Please, Professor, join us in welcoming Professor.
Thank you very much. I gave my presentation yesterday. So today, the professor in Spain, the director, he shared this presentation with me. So I want to share this one with you because They are hiring some PhD postdocs. There are 12 professors in the group. They have local and international projects, and they have money, and they invite some students to work in our group. So I will give little information, and there are some catalytic people. So can we open after this one? Can you open my presentation from yesterday? I will mention just 5 minutes about it. Okay, this is the group I joined 2 months ago, biorefinery process group. It's in Spain, San Sebastian, and in the Basque Country, University of Basque Country, there are 3 different campuses, and one is a campus de Vizcaya. The names are difficult for me, and there are 3 campuses and 31 faculties, 5,000 staff, and 45,000 students. And Faculty of Engineering, we are working here in this building. And this is the group members, not all of them, I am not here yet. And there are 12 professors. Dr. Jalal Labidi, he is the group leader, and 2 postdoctoral researchers, 5 PhD students, and we have many Erasmus students right now. And many exchange students are coming during the summer. And what we are doing, we are working on biomass fractionation, building blocks from polysaccharides, lignin depolymerization, extraction of bioactive molecules and their applications, new applications of bio-based materials, biocomposites, chitin and chitosan-based materials, I like these things, and materials for energy storage, process intensification, and process design and life cycle analysis. And the equipment, we have a lot of things. I will put all of them here. Reactors, we have very big reactors in our lab. Ultrafiltration equipments, homogenizer, and incubators and pilot plants, and liquid chromatography, HPLC, GPC, gas chromatography, spectroscopy, contact angle, and TGA, DSC, mechanical, optical, and AFM light. We have everything. If we don't have things in our lab, we use the other labs in the Bilbao. So almost we have everything there. And this group has been known from the Basque government from 2012. There are 30 PhD theses have been done and 300 publications, 21 book chapters, and 45 research projects. Almost all the professors, they have projects in the group, and contracts with companies, 20 contracts they have, and international patents too, and coordination of network excellence on sustainable biorefineries and research centers. And some selected papers he put here. And smart films, they are making films with lignin, with chitosan, chitin, any kinds of bioproduct, they are making film. And they are making chitin, chitosan from different kinds of animals, and then making bioplastics. And the same, they are making hydrogels from chitin nanofibers and using this one for different kinds of applications. And this group is very good for lignin extraction and the application of lignin. And lignin hydrogels, they are producing lignin hydrogels. And this is the city, San Sebastian, and the university is somewhere here, just 2 minutes from this beach. And this is full of tourists during the summer. During the winter, I was there last month. Many people are coming from USA, from Europe, and this is the city center. So some view from the city, as you see, and this is all about the lab. This is the professor. If any of your students, or if you are interested in cooperation, you can apply. You can come with our projects, or your PhD students can come for postdocs or anything else. Please contact with the Professor Jalal Labidi directly, or you can write me directly. I can discuss with him. So this is all for this one. Can we go the other one, please? So I have many materials, but for one material, I think it can be used for catalytic reaction. So I want to share that one. I will go a bit quickly, sorry, to find the correct one. This one, yeah. So this is the sponge. We are working with a professor from University of Copenhagen. They have, there is a museum named Natural History Museum. So there are a lot of animals and plants. So this samples from the Natural History Museum in Copenhagen. And here in the sample, there are the silica fibers and biosilica beads. I separated both of them by chemical reactions. And then they are the biosilica beads. When I was getting the SEM images, all of them, the out surfaces like this is closed. But I faced one of them like this. I thought that is that different because there are the pores here, but here there is no pores, it is closed. So I decided to break it. When I break this biosilica beads, there is a hole inside. Do you see the hole? There is a hole inside. Normally the outside is closed, but there are the channels. These channels are coming until here, but not opening, not reaching outside. So we use hydrogen fluoride to melt the surface, and we reached porous biosilica beads like that. We tried many methods, and then we made a standardization. There are holes inside. All these channels are opening the hole inside, and there are about 3,500 pores opening the gap inside. So what I did, this is negatively charged. It absorbed all the cationic dyes very well. And when I read the biosilica bead, it is active or not active. If it is bioactive, if you put it in the simulated body fluid like blood, it produces hydroxyapatite. Hydroxyapatite is the main component of the bone. So when we put this one into the hydroxyapatite, put into simulated body fluid, it produces hydroxyapatite, as you see. So it's good for bone tissue engineering. Then I use it some osteoblast cells. The osteoblast cells can grow on the surface, and the cells look very happy, and they attach from one beads to other beads. This is great for us. So for the bone tissue engineering, we need two things, hydroxyapatite and the bone cells. We have both of them. So this can be used as a bone tissue engineering material. Also, we can load any drugs inside. Then this is the summary. We extracted the beads and then hydrogen fluoride treatment. Then we reached porous biosilica beads. It produces hydroxyapatite, and the osteoblast cells can grow on the surface. Then we published this one in Chemical Engineering Journal 2 years ago. So it's on the market. We are selling this one, 1 gram for $100. It's just for laboratory experiments. And this is the material. So can we use this one for catalytic reactions? Please think about that if you're interested, or if you have any other idea, because we gave a name this material like giant macroporous silica. So it can be used for any other applications. There is a professor in Harvard University. Her name is Johanna Eisenberg, and he's finding one biosilica beads and publishing maybe 30 papers in Science and Nature. We are not like her, but I want to publish. We published only one until now, but we want to publish more papers with this material. So that's all today. Thank you very much for joining.
Thank you, Professor Kher. And now we will proceed to the closure of the. Are there any questions for the professor? Yes, there is.
Mechanical stability means like young modulus or something like this. This is a biosilica amorphous, so it's not very mechanically strong. But still, it has significant real mechanical effect.
I think Professor Tomic, that professor from the Institute of Vinča, Belgrade, he talked something similar, but with the synthetic hydroxyapatite. He trying the same things, but the synthetic hydroxyapatite. My recommendation is that Professor Tomic sent to you the contact of the professors from the Institute of Vinča. I think it will be very valuable to compare that natural source and natural materials with that synthetic, and maybe finally something as a solution for the problems with the bones, especially cancers, the bones, tooths, with the, how call it, when we treat the medical tooths, that professor explained, it will be a completely new generation of the recovery of the tooths if that technology come on the line.
This would be great for me. I
So
want to
it will be
contact with him.
good to make the contact.
Yeah.
Both of you.
I will do that.
Yes, thank you. Are there any more questions?
Thank you very much.
Thank you. Thank you once again. And now we will proceed with the closure of this event. And I give a word to our dean, Professor Dragan Vedinovic.
Thank you. Thank you, Milan. Compared to the opening ceremony for the closing, I have only one page. And I must say that I'm so sorry because our second invited lecturer, Dragana Tomašević-Filipović, had accident during the skiing, and I wish to have fast recovery process as much as possible, easy process. You know, accident happened, and I'm so sorry because of that. Dear guests, here we are at the end of the conference EEM 2023, so it is time for us to make some concluding remarks. Compared to the data from the previous events, we can conclude that the EEM 2023 event had an excellent turnout, judging by the number of submissions and the attendance of authors and co-authors. The number of attending institutions was also remarkable, as an efficient number of submissions were related to the problems and topics defined by the national strategies and international projects. The turnout of young researchers was also sufficiently high. All plenary speakers kindly accepted the invitation and held their lectures. All invited speakers also accepted the invitation, and out of 12, 10 were held. We can conclude that EEM 2023 event was successful, judging by the fact that once again we did manage to provide both on-site and online participation and achieve the goals we had set. Ladies and gentlemen, dear participants, bringing this event to the conclusion, on behalf of the organizing committee on the Faculty of Technology, Zvornik, University of East Sarajevo, I would like to express our sincere gratitude to all the authors and co-authors for their submission, to those who attend the gathering both in the person and virtually. And we are also especially thanks to those who have been with us from the first EEM back to 2009 and to 2009, to our esteemed plenary speakers and invited speakers, members of the scientific and organizing committees, associates, to reviewers, sponsors, our technical organizer, Happy Travel, to our technical support. I will probably talk about any technical problems which we may be. To the media and our host, the Kotel Termak staff and management. We wish you all safe journey to your homes. With this, I pronounce the 8th International Congress in Genomic and Environmental Materials and Process Industry in 2023 finally closed. Thank you.